Well, we have four big objectives today. First is to understand the spectrum of genetic variants that affect height. The second is to be able to differentiate between polygenic short stature and monogenic short stature, and I'll talk a little bit about what those terms mean. Next, we're going to review a few important genetic causes of short stature that affect the growth plate. We're going to pay particular attention to two, the Aggrecan gene and the NPR2 gene. And finally, we'll step back and discuss the application of genetic testing in the workup of patients with short stature. So when I think about an individual person's height, I realize that there are a number of factors that underlie how tall someone's going to be. So pictured on the right of the screen here are factors that affect the patient's environment, the individual's environment. So obviously their nutritional status. If a child is significantly undernourished, they won't grow well. We have there a bottle of growth hormone or any other medication. We know there are many medications that can have effects on growth. For example, corticosteroids, glucocorticoids, they classically will inhibit an individual's growth. If there's stress, psychosocial stressors can definitely lead to short stature and growth retardation. On the bottom is pictured a child with congenital hypothyroidism that has been untreated, and that has both genetic influences as well as environmental influences. And that's just a reminder that there are many chronic diseases that can have significant impacts on an individual child's growth. In the top, you have the picture of the in utero environment. So again, here there are genetic and environmental influences as well as epigenetic influences, where what the child, what the fetus experiences in utero can have long-term effects on that individual's growth. But today we're going to focus purely on the genetic aspects. So pictured here on the left is a picture of Muggsy Bogues and Manute Bol, who at the time were the tallest and shortest players in the NBA. And what we know is that within an individual population, approximately 80 to 90% of the variability in height is due to genetic factors. Now, as a pediatric endocrinologist, when I did my training in medical school and even in residency and fellowship, this was really how we were taught to think about growth, right? So you have the brain, and right under the brain, there's the pituitary gland, everyone's favorite gland, which makes growth hormone. Growth hormone goes to the liver, which produces IGF-1, and IGF-1 then goes to the bone and makes you grow taller. And of course, growth hormone also has direct effects on the bone itself, and IGF-1 is bound in this complex to its binding proteins. And then there was this black box, which was the growth plate. And really, we didn't learn that much about it. And if you were being particularly sophisticated, you realize that under that black box, there were these cells called chondrocytes, which had to differentiate and proliferate in order to make the bones grow longer. But the education was really focused on this hormonal axis and its influence on growth. And you know, I think that there are two adages that I like to think about that really, I think, well describe the diagnostic and therapeutic approach in the era of growth hormone. And, you know, I say this with a little bit of a joke, but also I think that there's some real truth behind these adages. So the first is the story of the man who a man's walking down the street and he finds another person who's hunched on his hands and knees under the light, under a lamppost, searching for something. He says, "Oh, did you lose something? Can I help you?" And the man says, "Yes, I dropped my keys." And the first man says, "Okay, well, is this where you dropped your keys?" And he said, "No, I actually dropped them down the block over there." So the man turns to him and says, "Well, if you dropped them down the block over there, why are you looking for them here?" And he said, "Well, this is where the light is." Right? So I think that's very appropriate in that for many years, we really only understood the hormonal regulation of growth. We learned a lot about the growth hormone IGF-1 axis and about the endocrine physiology of growth. And that was really where the light was in our field. So we looked at growth from that perspective. We looked at growth in trying to understand how those hormonal etiologies applied. And the second adage is, of course, when all you have is a hammer, everything looks like a nail. And of course, therapeutically, we really only had for many years growth hormone and then, of course, IGF-1. But that was our primary treatment modality for individuals with short stature. So we, of course, looked at the problem through that lens of growth hormone treatment. And now, in the last decade, the power of genomics has come and has shined a whole new light on growth biology. And I'm going to come back to this slide in a second, but we've learned that thousands of genes are involved in growth, including many different pathways, not just the growth hormone IGF axis. So before we get back to what that slide is showing us, I want to get us all on the same page to understand what we're thinking about when we talk about genetic variants. So when I think of any specific genetic variant, right, any change that makes each and every one of us unique, I think about two key descriptors of that genetic variant. So you may know that our genomes have approximately 3 billion base pairs, and we all have some differences. Although we're 99.9% similar, there's still millions of differences between each of us. And each one of those differences can be described as having what's called an allele frequency and an effect size. So on our x-axis, you see allele frequency. And what that means is how common is this genetic variant within the population? Is this something that happened just in that one individual, in which case it's very, very rare? Is it in 1 in a million people? Or is this something that maybe half of us have, or 5 or 10% of us, in which case it's pretty common? And then on the y-axis is the effect size. And that means how much of an effect does this individual genetic variant have on our disease or trait of interest? So if it were a disease, for example, like your risk of type 2 diabetes, does this increase your risk by 5%, or does it increase your risk 50-fold, right? That would be a huge effect compared to the small one. So with respect to height, does this individual genetic variant change your height by just a millimeter or a few millimeters? Or does it make you, a foot or 2 feet smaller? So in the top left corner of this graph, we have rare genetic variants that have large effects on height. And those are what we classically call monogenic disorders, where there's a single gene or a single genetic variant or a few variants in a specific gene that is causing you to be significantly smaller. So the classic example of this would be achondroplasia, right? Where you have one specific genetic change in the FGFR3 gene, and as a result, you're significantly shorter, -5, -4 standard deviations compared to the mean. And then all the way on the bottom right, we have common genetic variants that contribute to what we call complex disease or traits. Now, height is a complex disease or trait, which means that these common variants, there are literally thousands of them that are common in the population. Maybe 5%, 10%, 15% of us have them, but each one only contributes millimeters to our height, but in aggregate, they'll have a significant contribution. And then in the middle are these low frequency variants that might have moderate effect size. So these common genetic variants we identify using a tool called a genome-wide association study, or a GWAS. And I'm going to explain more about what that is in a second. And the rare genetic variants are what we're doing when we do sequencing, when we are doing clinical testing in the office to try and find the cause for an individual's short stature. We're often looking for one of these monogenic disorders. It's much harder to find those ones in the middle where you really need to sequence lots and lots of people to figure them out. We're not going to really focus on those today. Okay, so what is a genome-wide association study? So I think people are pretty familiar with the idea of an association study, right? An association study is when you take a group of individuals with a disease and then a group of controls, then you see whether a specific risk factor is more common in those with the disease as opposed to the controls. So let's use our diabetes example for a second. So we have 100 people with type 2 diabetes and 100 people without type 2 diabetes. And we might say, okay, we're looking at one specific gene or genetic variant to see, is that genetic variant more common in the individuals with type 2 diabetes than without? And if 90 out of the 100 have it, and the people with diabetes and only 5 people have it without a diabetes, then you get an odds ratio, and you would see that, yes, of course, that is increasing your risk for having type 2 diabetes. Now, you can also do this for what we call quantitative traits. So traits where you actually have measurements. It's not just do you have it or do you not have it. So for example, height. And in this case, you would say, okay, does that genetic variant on average make individuals taller? You can do a regression analysis. Now that's a typical association study. So the only difference between a typical association study and a genome-wide association study is that instead of doing it for one genetic variant or one risk factor, you're now doing it for millions, right? So you're doing it for looking at kind of common genetic variants across the entire genome. Often, a few million what we call SNPs, or single nucleotide polymorphisms. So we're looking at the common changes where 10% of us might have an A and 90% of us have a C, and we look at those across the entire genome. And what that does is it doesn't tell us, is that specific genetic variant the cause of you being taller or shorter, but rather what it does is it identifies areas of the genome that are associated with height in the general population, right? Where there's a signal where something's going on in that area of the genome, and then you can try and figure out which gene it is and which variant's actually causing it in that signal, in that area of signal. Now, this has been done now in very, very large genome-wide association studies. This is one of the more recent ones, which had 700,000 individuals who had this type of genotyping done across their entire genome, and we had height data on them. This was done by the Giants Consortium, which my mentor, Joel Hirschhorn, is one of the key investigators as part of that. And they found that they've now found 700 different regions around the genome that are significantly associated with height in the general population. And again, each of these regions has common genetic variants that are, each one may only contribute a millimeter or a few millimeters to height. But when you start adding up, there are 3,290 significant variants in those 712 regions. You can start to explain a significant portion of the variation in height. And it's estimated that just adding up all these common genetic variants, you might even explain 50 or 60% of the variation in height. Now that you have these thousands of variants in hundreds of different regions across the genome, you can start doing pathway analysis to look at what are the genes involved in those specific regions. And when you do that, of course, you do find the growth hormone and IGF pathway, which is very reassuring. If it wasn't there, we'd have to scratch our heads and make sure we're doing the right thing. But we start to find all of these other pathways. And the ones that I've highlighted in red are all pathways that are involved in the growth plate, in bone and cartilage development, which makes sense as that is the area that actually makes you grow. And we're starting to appreciate that there's this concept of a range of genetic variants, like I showed you in that initial graph with the X and Y axis, where you have rare genetic variants that have severe effects, and those are ones that lead to the monogenic disorders, the skeletal dysplasias. But then you can have more mild variants in the same genes that might lead to isolated short stature, and even more common variants that might just add that millimeter or subtract that millimeter in the general population. And on the flip side, actually, you could have variants that are the opposite effect that might actually make you taller as well. So this is an important concept. And what polygenic short stature means is polygenic short stature means that that there's not one cause of you being taller or shorter, but it's this aggregate add up of your hundreds, if not thousands, of common genetic variants that are determining how tall you are. So here's one area I'm going to zoom in just to give you an example. This is one of the most associated regions in the genome. So on the y-axis on the left side here, you see what's called the -log10 p-value, which means that these associations are significant at 10 to the minus, and you see up to 30. So that means you have a decimal, a 0.000000, keep going 30 times, right? That's how significant these associations are. And what you see is there are multiple significant associations clustering in the middle here around the gene called Aggrecan. All right, so remember that for a second. We're going to come back to this gene. But each of these associations has, again, millimeter effects on height in the general population. Now, back when I was at Boston Children's in 2014, I collaborated with Ola Nilsson and Jeff Barron and a number of other investigators and physicians, and we identified these 3 families. And if you look at the pedigrees of these families, you'll notice that the short stature is inherited in a dominant fashion, meaning that there's one parent and then one grandparent. You can trace the short stature back in the family coming from one side of the family. And what was remarkable about each of these children who showed up to our clinics was that all 3 of them had advanced bone age x-rays. So I think for the endocrinologists in the audience, you're all pretty familiar with measuring a bone age x-ray to look at the maturation of the growth plates. And the vast majority of the time, patients who present with short stature either have delayed bone ages or maybe bone ages similar to their chronologic age. But it's extremely unusual to have a bone age advanced where the bones actually look older than the individual. And when we investigated these individuals, we saw that there was a range of short stature from around -2 to -4 standard deviations. And the bone age were advanced between 1.5 and 4 years. And what was truly remarkable was that 2 of the mothers reported that they had stopped growing prior to the onset of menarche, right? Prior to them having their first menstrual periods. Now that is not normal human physiology. And they had normally timed puberty. And there was no hormonal reason why the bone age should be advanced. They didn't have any increased androgen levels, any increased estrogen levels. There wasn't anything to really explain that bone age advancement. And one of the families had early onset significant arthritis and osteochondritis dissecans. And we did a whole exome sequencing, and we found that all 3 families had heterozygous mutations, meaning that one copy of the gene had a mutation in the Aggrecan gene. So Aggrecan is a proteoglycan. It's found in the extracellular matrix in the growth plate and also on the articular surfaces of cartilage. And prior to our publishing this paper in 2014, there had been 3 families in the world ever described with mutations in the Aggrecan gene. And each one of them had their own skeletal dysplasia named after them. So what we learned from this study was that we had found the tip of the iceberg with those initial skeletal dysplasias, right? Those were the most severe cases that were found with Aggrecan gene mutations. And now we're starting to move down that iceberg, and we found that, oh, in fact, you can present with isolated short stature with advanced bone age. And then once we started talking about this in 2014, we then reached out to investigators all around the world, and we were able to put together now 103 individuals with Aggrecan mutations from 20 different families. And what you see in red is that around half of the families had significant early onset joint disease. And we also learned that the bone ages and the degree of short stature was really even more variable. So not everyone was as severely affected as those initial 3 families. So here's a graph showing on the right is the growth chart for boys and on the left for girls, showing the degree of short stature and the bone age advancement, which was highly variable. And there have been even a few cases reported now of patients with clearly pathogenic Aggrecan mutations who didn't even have an advanced bone age, although I would say that's the exception and not the rule. And now there have been multiple other papers. I've just put 2 here. One from showing a group of patients who were born small for gestational age with advanced bone age, that 14% of them had Aggrecan mutations. Another looking at children with short stature and mild skeletal defects, also showing that around 10% of those had had Aggrecan gene mutations, slightly less. So, and now there literally have been hundreds of patients found with mutations in the Aggrecan gene. And this is likely one of the more common causes of at least dominantly inherited short stature, an idea we'll come back to a little bit later. When I was at Cincinnati Children's, I launched a clinical trial in collaboration with Dr. Bacaliol, who will be the seminar's next speaker. And this was supported by Novo Nordisk, where we did a trial of growth hormone therapy, again, with it being really the only available therapy to treat these patients, to see if in a standardized prospective trial, we could improve height outcomes in those patients. And this is preliminary data, which has been only published in abstract form, that shows that of the initial 6 patients who completed a year of therapy, the median height velocity was 8.9 centimeters and a median height standard deviation score of 0.6. So pretty good for the first year. And it didn't seem to lead to an unwanted advanced bone age in excess to what you would expect from the Aggrecan mutation. But what was also remarkable is that we decided to do some detailed phenotyping of these kids as well. So to look at the involvement, the joint involvement. And 3 of the children were found to have osteochondritis dissecans on their knee MRI, despite reporting no symptoms. And many of the parents had severe early onset arthritis. So this really showed us that the pathology at the joints can start at quite a young age and even before anyone presents with symptoms. And perhaps we need to start thinking of altering these kids' activities to try and prevent, long-term degenerative joint disease. But more research is needed to be done to really figure out what is the best approach for these children. So the take-home point that I wanted to get to you from this Aggrecan story is that there are variants in that same gene that have a range of effects across the spectrum. So you saw from the Genome-wide association study that Aggrecan influences height in the general population by just a few millimeters, but then there's going to be this range all the way to severe skeletal dysplasias. And that many of the genes involved in growth are not necessarily classical endocrine genes, but rather skeletal or growth plate genes. So with that idea in mind, I want to turn and focus a little bit more on the growth plate. So pictured here on the left is a zoom in of the growth plate. This is taken from an excellent review article by Jeff Barron that I would point you all to in Nature Reviews Endocrinology. And what it shows you is the columns of the chondrocytes, which are in different zones, the hypertrophic zone, the proliferative zone, the resting zone, and then at the bottom, you see here the new bone being developed. And if you zoom in on the chondrocyte cell, so on the right now, we've zoomed into one cell, there's one particular signaling pathway that I want to spend a little bit of time here. So people are probably familiar. Here is the FGFR3 gene. So that is the gene that when it has activating mutations, it leads to achondroplasia or hypochondroplasia. And what that does is through these signaling pathways pictured here, and particularly this MAP kinase pathway, the activation of FGFR3 inhibits chondrocyte proliferation and hypertrophy. So really leading to aberrant growth plate, physiology and impaired growth. So you might notice that there's some other parts of this pathway that might look familiar to you. So this RAS, SAS, RAF part, that's those are the key molecules in Noonan syndrome, or part of the key molecules in Noonan syndrome, and in a class of disorders called restopathies, which we'll come back to. But now I want to draw your attention to the top right, to this CNP-NPR2 component. So CNP stands for C-type natriuretic peptide, which binds to its receptor, the natriuretic peptide receptor type B, which is encoded by the NPR2 gene. And the purpose of this pathway is actually to promote growth. So as you can see, this pathway actually intersects with the FGFR3 pathway here, and it inhibits that pathway, thereby actually leading to promote chondrocyte hypertrophy and increase growth. So why is this pathway important? Well, back in 2004, it was found that individuals with recessive mutations in the NPR2 gene, that's that receptor for CMP, they have a significant skeletal dysplasia called acromesomelic dysplasia maritotype. So very severe. But what was interesting was that a group led by Rob Olney realized that the parents and siblings of these individuals who had heterozygous mutations in this gene, they ended up having what looked like idiopathic short stature. So they were shorter than the family members who didn't carry one copy of this mutation. But on X-ray, they looked otherwise like they didn't have any significant skeletal dysplasia. You wouldn't be able to realize that they had an underlying skeletal abnormality. And following that, there have been multiple groups. I've just put up 3 here, which have looked at how common heterozygous variants in the NPR2 gene are in idiopathic short stature. And I would say the consensus is that around 2% of idiopathic short stature is caused by mutations in the NPR2 gene. So that's pretty common as genetic causes go. And what was really interesting is on the flip side of this, there have now been 4 papers, one of which was just published in July, showing activating mutations of the NPR2 gene. So these are where the that receptor is turned on and thereby continues to promote growth. And these individuals in these 4 families all have significant tall stature. So pictured here is in the middle is an individual whose height was 7 feet 3 inches. And the doctor standing next to him from the Netherlands look like they're small, but I actually think the male doctor is over 6 feet himself. But it's just because this individual in the middle has such significant tall stature from his activating NPR2 mutation, showing that if you have increased signaling in this pathway, you can promote growth. Now, we and others asked the question, if this NPR2 gene, if you only need one copy of it to cause short stature, you would think that, again, it would follow a dominant pattern where that one copy was inherited from one of the parents, and there would be a history of familial short stature. So looking at our study in Boston of patients with idiopathic short stature, we pulled out the 99 children where we had height data available on both parents. And of those, 22 of them, so around 20% of them, had one parent who also had a height below -2 standard deviation. So we'll call that familial short stature. And now these are small numbers, but of those, 3 of them had NPR2 mutations. So 14% of the familial short stature had NPR2 mutations. Interestingly, before we get to the second study, only one individual out of the ones without familial short stature also had an NPR2 mutation. And that was a de novo mutation, meaning it was a new mutation that neither of their parents had. So it is quite rare to have an NPR2 mutation if you don't have a family history of short stature. Now, in a somewhat larger study out of the Czech Republic, 917 children with short stature, they had 12% who had familial short stature, also defined as a parent with short stature, one parent with short stature. And of those, around 5% had NPR2 mutations. So I don't think we know the exact number, but you see that in families where one parent has short stature, this as a cause of dominantly inherited short stature is going to be enriched. So NPR2 is going to be an important player. So why am I highlighting this one cause? Because I'm going to mention a number of other causes in a minute. The reason I'm highlighting this cause is because now a number of companies are developing novel therapies to target the NPR2 pathway. So I'm just showing you one study. This is a study that was published in the New England Journal last year. It's a study of a drug called Vosoritide, made by a company, BioMarin. And again, as my disclosure, I have funding from them as well, although I was not involved in the study in any way. So this study was looking at Vosoritide, which is an analog of C-type natriuretic peptide. And what they found was that in patients, they did this, this was a phase 2 study in patients with achondroplasia. Again, if you'll remember from that pathway, FGFR3 is activated in achondroplasia, and now you're using CMP to inhibit that activation and promote growth. And what they did was they found a sustained increase in growth velocity in patients receiving Vosoritide. And actually, this past Friday, they just published their phase 3 study results in The Lancet. I didn't have time to make a newer slide, but that was a randomized controlled study trial of Vosoritide versus placebo in patients with achondroplasia, and also showed an improvement in growth velocity in patients who receive the Vosoritide. And there are other companies as well. Ascendis is making a transcon CNP as well, a longer acting CNP. And there are a number of other approaches that are looking at the use of C-type natriuretic peptide as a potential therapy for achondroplasia, where this axis is specifically perturbed. Now, I thought that this CNP molecule could be helpful for many other causes of short stature. We have recently here at Children's National launched the first study looking at Vosoritide in any patients outside of achondroplasia. And specifically, we're looking at 5 disorders for which it makes sense based on the molecular signaling pathway that Vosoritide or a CNP analog could be helpful. So we're recruiting now patients with hypochondroplasia, with any type of restopathy, so that's Noonan syndrome, neurofibromatosis type 1, Costello syndrome, cardiofaciocutaneous syndrome, some other rarer ones, also involving that pathway. Individuals with SHOX deficiency, because that seems to also interact with the CMP NPR2 pathway. And then, of course, patients who are missing one copy or have a defective copy of the NPR2 mutation, the NPR2 gene, and then those who don't produce CNP, which is a very rare disorder. So we're currently doing a phase 2 study of Vosoritide in prepubertal patients who have to live in the United States with any of these 5 disorders. And the design of the trial is that they come for a screening visit here to Children's National. We do a 6-month observation period where we're just measuring their baseline growth velocity, and then we'll treat them with Vosoritide for a year. So if you have any patients you think would be interested or are eligible, please feel free to reach out to me. My email is adauber@childrensnational.org, but please feel free to reach out to me. We'd be happy to talk to them about potentially being involved in the trial. All travel and costs of the study are covered by the study budget, and this is being funded by BioMarin. Okay, so while we focused on NPR2 and Aggrecan, there are many other genes which, when we're doing our genetic testing, we're realizing are subgroups of what was formerly called idiopathic short stature. So I think that genomics over the years is going to try and take the idiopathic out of short stature, right? So Aggrecan, NPR2, each of those are probably around 2% of short stature, but there are also, of course, the SHOX gene, Indian hedgehog, Noonan syndrome, FGFR3, and many, many other genes. And the mutations in these genes present with a wide range of short stature, and some of them can have significant associated medical comorbidities, like the arthritis in Aggrecan, or of course the cardiac involvement in Noonan syndrome and other syndromes. All right, now I've been focusing on genetic sequence variants, but I just want to take a second to realize that there are other types of genomic variants also that can have a significant effect on short stature. And this was a study I did back in 2011, the details of which I'm not going to go into, but what this was looking at whether individuals with short stature were more likely to have larger genomic deletions as opposed to individual sequence variants. And what we found was, yes, that individuals with short stature had a larger burden of rare genomic deletions, larger deletions. And a number of groups have now looked in patients who come with short stature, and especially if they have additional syndromic features, up to 10 or maybe even 25% of those patients might have genomic deletions, also called copy number variants. They can also have duplications underlying their short stature. And that's where the role of a chromosomal microarray may come in the evaluation, especially of syndromic kids or kids who also have associated intellectual disability or other symptoms or features. And finally, I'm going to summarize all of epigenetics in one slide. So there is something called epigenetics, which is not changes to the individual sequence of the gene or whether the copies of the gene, but changes to attachments to the DNA or kind of the overall structure of the DNA. So the most common things we think about are methylation, DNA methylation, but there are other things as well, histone marks and other things. And there are genes that have a phenomenon called imprinting, where only one copy of the gene is expressed, depending on whether it's inherited from the mother or the father. And this is typically controlled by methylation. And the classic growth disorder that is involved in this is Russell-Silver syndrome, or for those of you in Europe, Silver-Russell syndrome. That is due to an epigenetic, either an absence of methylation. There are a number of different causes of it, but epigenetic changes. So I just want to note that there are actually multiple growth disorders that have either imprinting defects or epigenetic defects, and there's probably a lot more to be learned on the impact of epigenetics on the whole field of growth disorders. So stepping back, this is my overall kind of view of how I think about growth and the genetic causes. So on our top left is the height distribution in the general population. And at the extremes of the general population, you'll find individuals, many individuals, who have that polygenic short stature, where there are lots of different genetic variants that just in aggregate are leading them to be towards the lower end of the spectrum. But there are also individuals in there who might have rare genetic variants, giving them a monogenic cause. And if you just pull out all of the individuals with idiopathic short stature, you'll see there's some individuals with the blue cause or the green cause or the orange cause or the red cause, which may be NPR2 or SHOX or Aggrecan. And those individuals also are going to have variability in their degree that they're affected. And further, if you pulled out the families where there's just a clear pattern of dominantly inherited short stature, those families are going to be even more enriched for having the monogenic causes. So this comes to what I call my mid-parental height tangent. So I think most of us are familiar with the concept of the mid-parental target height, which is the idea is if you take the father's height and the mother's height and you average them out, and then boys you add 2.5 inches, girls you subtract 2.5 inches, and that is on average where their child should end up. And that whole idea is based on polygenic stature, right? That whole idea is based on the idea that, sure, there are thousands, hundreds or thousands of common genetic variants, each of which have a small effect on the kid's height. But in aggregate, if you get half from your mom, half from your dad, and you get, you mix them all up together, the child will end up somewhere in the middle of that expectation. Of course, and there's going to be some variability between siblings depending on what mix they got. But this concept of the mid-parental height does not make any sense when you apply it to monogenic short stature. So, for example, let's just take an extreme example. If you have a father who has achondroplasia and a mother who does not have achondroplasia, let's pretend that the mom is 6 feet tall and the dad is 4 feet tall. So achondroplasia is a dominantly inherited condition. Each child has a 50% chance of having it, right? Their children will not end up 5 feet tall, right? They're not going to end up smack in the middle of the shorter father and the taller mother. No, their child will either have achondroplasia or will not have achondroplasia. And that's going to be the overwhelming determinant of their height. And if they have achondroplasia, they're going to have a height similar to their father. And if they don't have achondroplasia, they'll be much taller than that 5-foot average, right? Because that one genetic variant is really overall determining it. And if you don't have that variant, then you fall back to the rest of those polygenic variants. But what's really confusing and what really happens in our clinics all the time is that you have this mix, right? It's not as clear cut as achondroplasia or not. Rather, one parent might have a variance like an NPR2 or something, which might drop them by one standard deviation, but then there's a whole bunch of polygenic variants which might make them taller or shorter. And you need to kind of take all of these into consideration and see where the kids are going to end up in between. Did they get that, larger effect variant or not? And how does that average out with all of the small effect variants? So the clinical take home is that it's important to look at the pedigree when you're considering a genetic etiology of short stature. And you need more information than just the parents' heights. You really need to look at the siblings. You need to look at multiple generations. And if you have multiple generations with short stature on one side of the family, you should think of a dominant condition. And maybe we're more likely to find a genetic cause there. And if you have very discordant heights between siblings, that also would favor potentially a monogenic condition. All right, so in the last few minutes, how do we apply this clinically? So the question that I often get is, should we do genetic testing for patients who come into clinic with short stature? And I think the answer is, it depends. It depends really on whether you think they have a monogenic condition. There are clear benefits to genetic testing, right? If you are able to find a specific diagnosis, that alone for many families is very helpful to stop ordering lots of other tests, stop doing other x-rays or a whole diagnostic odyssey. To be able to put a name to what's going on in their family can be very helpful. It can also be helpful for prognosticating. For example, if you knew a patient has an Aggrecan gene mutation, those kids tend not to have a robust pubertal growth spurt, and the height standard deviation worsens over time because of the premature growth cessation. That helps you counsel families about whether therapy would be helpful. Of course, it also can tell you about other associated comorbidities. It also allows you to do testing within the family to find out who else might be affected. And there may, in some instances, be treatment implications. I didn't have time today, but we found some patients with dominantly inherited mutations in the growth hormone receptor who may do better with IGF-1 treatment, or we'll see the results of our trial. Potentially there'll be other precision therapies that can help with individuals with NPR2 gene mutations or other specific mutations. So let's move on. So the first question you have to ask yourself is, do you think your patient has a monogenic disorder? So the clues for that are, what's the severity? Are there other associated congenital anomalies? Were they born SGA? Do they have dysmorphic features? Is there something very unique about their biochemical phenotype? For example, is their IGF-1 level very high and they were born SGA with microcephaly? You should think about an IGF-1 receptor mutation. Is there those familial patterns that we talked about? If there's consanguinity, perhaps there's a recessive disorder if multiple siblings are affected. These are all clues where you think that perhaps there's a monogenic condition where genetic testing would be helpful. If both parents are, between -1 and -2 standard deviations, and everyone in the family is around the, 3rd or 5th percentile, and this child's growing at the 3rd or 5th percentile, I think that you're very unlikely to find a single genetic cause, and genetic testing probably won't be so helpful. And then, if you look at the patient, this is where a clinical history and physical exam are key. You still need to look and see, are there physical features suggestive of a specific candidate genetic syndrome? And if there are, you should do testing for that specific syndrome. So for example, I was recently in clinic with one of our fellows, and a child came in with significant short stature, actually had already a whole endocrine workup. And I looked at the child, we measured their body proportions, and she was significantly disproportionate. And when I looked at her mom, I noticed that her mother, who was also very short, had a madalung deformity. We did SHOX testing, and lo and behold, she had a heterozygous deletion of the SHOX gene, giving her Léry-Weill dyschondrosteosis. So if there's a clinical candidate syndrome, you should do the testing for that specific syndrome. However, if there's no candidate, but they just have unexplained short stature, this is where doing a larger genomic evaluation, like a chromosomal microarray or exome sequencing, can be helpful. And just to show you some evidence for that, this was a well-done study from Hauer et al. It was published in 2018. They took 565 kids where they did what they called systematic phenotyping. And this was really, they came to a growth clinic where there was an endocrinologist and a geneticist, and they did kind of a targeted workup, including chromosomal microarrays and targeted testing for patients who they thought had a specific condition. They were able to identify a genetic cause in 14%. They then took 200 of the patients who didn't have a specific cause and did exome sequencing in them, and they found an additional diagnosis in a 16 more percent, giving a combined yield, if you do the math correctly, of around a third of the patients could be genetic diagnosis could be found. And if you look at that, some interesting points: around 2% had mutations in Aggrecan, around 1.5% had mutations in NPR2, and almost 2/3 of the variants that were found followed a dominant inheritance pattern of exome sequencing. They were primarily in skeletal genes. A different group, this is the group from the Czech Republic. They did a study looking at severe familial short stature, so heights below -2.5 standard deviations in both the patient and their parents. And they found a genetic cause in 52% of those patients. And again, most of them were also in growth plate related genes. So I'm going to end though with one true case example, because I don't want to give you the false impression that genetic testing is a panacea and will always give you the answer. So this was a 9-year-old male whose height was at the 0.7 percentile. It's proportional, a little bit of a prominent forehead and triangular face, was born small for gestational age, but caught up by age 2, prepubertal. The bone age was a little bit delayed. The mother's height, you see there, 5 feet tall, the father 5'10". So the mom a little bit on the shorter side, but not outside the normal range, had a normal IGF-1 and BP3, and their primary care physician ordered whole exome sequencing. I cannot make this up. This is true story. And they came with a result from their exome sequencing to the pediatric endocrinologist, which was not me, and they asked them, what do you think of these results? So this is gonna happen to you, right? And this is what the report said. The report said that there were 2 maternally inherited heterozygous variants of uncertain significance. Clinical correlation requested. Basically, you decide what there are. And one of them was a novel missense mutation in the Aggrecan gene, and one was a missense mutation in the IGF-1 receptor, which was present in like 1 out of a few 100,000 people. So are either of these the cause? I don't know. I mean, there's really no way to know right now. While, you know, the Aggrecan mutation is predicted by some computer programs to be damaging, the patient's bone age is delayed, not advanced, and the mom's not that short. So could it be a contributor? It could, but I'm not sure. The IGF-1 receptor, similarly, the IGF-1 level is in the normal range, although that can be seen in IGF-1 receptor mutation. So sometimes you're going to get results that are very confusing, and we really need a lot more research before we can really understand the meaning and the significance of all of these genetic testing. But sometimes you're going to get results that are very clear-cut, where it does give you a definitive answer. So in conclusion, the main lessons I want you to learn today are that variants in the same gene may have a range of effects across that spectrum of allele frequency and effect size, and that any individual's height is determined by the combination of many different genetic variants. And in those with monogenic conditions, they have a rare variant that has a larger effect, but that alone doesn't fully explain their height. Many of the genes involved are not classical endocrine genes, but are also primarily affecting at the skeleton and at the growth plate. idiopathic short stature is going to be subdivided into many new diagnostic bins and into this concept of polygenic and monogenic short stature. And genetic testing can definitely help establish a diagnosis, but at times it will also cause tremendous confusion. So I'm happy to answer any questions. Here is my email. Again, if you have patients who are interested or eligible for our study, please let me know. And we also are doing ongoing genetic studies for particularly interesting patients with unsolved cases of short stature. And with that, I'm happy to answer any questions.
Andrew, Ron here. Thank you for really a superb lecture. Very, very much appreciated. Let me kick off the questioning if I can. Andrew, do you envision a time in the foreseeable future when genetic testing will reach a point in terms of quality and cost where it will totally supplant customary biochemical testing in children with short stature? Will it replace growth hormone stimulation testing, HGF levels, etc.?
That's a great question, Ron. So my answer is for some patients, right? So there are patients who now come into my clinic where there's clearly a dominantly inherited pattern of short stature, there's clear disproportionate short stature, and in those patients, I honestly think that a genetic testing panel should be the first test and not the last test that is done. And those patients are more likely to have, mutations in the NPR2 gene or Aggrecan, where genetic testing is the test of choice. But then there are those patients who come in with, preserved weight and falling off height, where you're worried that this is growth hormone deficiency or hypothyroidism, or patients who haven't gained weight and are having GI symptoms where celiac testing needs to be considered. In those patients, genetic testing is, the biochemical testing needs to be the first line test, because a lot of causes of growth hormone deficiency, the yield for genetic testing will be very low. Or if you have a tumor, obviously you need to find that and not do genetic testing. So I think what I'm hoping is that we'll have a more nuanced approach to children with short stature, where the default isn't always to get thyroid studies and an IGF-1 and celiac testing and maybe a chemistry panel, but rather where you look at the individual patient's growth pattern, family history, and determine for yourself, is this more of a skeletal cause, in which case maybe I should do genetic testing, or am I worried more of a hormonal etiology where I'm going to do my kind of classic endocrine workup? But both of them have an important role.
Daphne, can you perhaps read some of the Q&A questions for Dr. Dauber?
Absolutely. Dr. Dauber, first of all, excellent presentation. We're receiving so many positive feedback. Dr. Rosenfeld, unfortunately, has to step away, so I'm going to take over the Q&A at this time. The next question we have is from Catherine Cutney. She's curious whether there are any genes that relate to puberty timing that impact final height in these GWA studies.
That's an excellent question. So yes, there are genes in the genome-wide association studies. There are genes around the androgen receptor, the estrogen receptor. There are signals that affect genes involved in pubertal development and pathway that underlie height as well. The truth is I don't know the answer of whether the same signals that affect pubertal timing, because there are other genome-wide association studies that have been done looking at the effects on pubertal timing. I don't know offhand if those same signals are the ones that affect final height. That's a good question, and somebody probably knows the answer, but I just don't know it offhand. But there definitely are crossover in the pathways between the two.
The next question is from Gabriella Stein. She wants to know what would be the mechanism explaining bone age advancement?
Great. So probably, I assume you're referring to the Aggrecan mutation, but there are a number of conditions, and what it has to do with, what a bone age really advancement means, is it means that the chondrocytes in the growth plate are prematurely differentiating, and they're kind of prematurely aging. You know, not really that they're getting older, but they're have this inherent program of, you know, how they should be progressing both in relationship to growth and puberty. And there are a number of genes that affect, and signaling pathways that affect that program. So Aggrecan, the exact mechanism isn't known. It seems to affect maybe some hedgehog signaling, some other of these signaling pathways that are involved in the growth plate and interactions between the matrix and the chondrocyte themselves. And those are kind of sending a signal to the chondrocytes telling them to advance their maturational stage and thus advancing the bone age. And there now have been a number of different genes that have been shown to have that effect. But the signals involved there are SHOX is involved. As I said, hedgehog signaling, BMP signaling, there are a bunch of different things involved.
Okay, great, thank you. A question from Oscar Escobar. In individuals with FHX of ACAN, can aromatase inhibitors have an effect on avoiding bone age advancement?
Yeah, so that's actually, thank you for that question, Oscar. That's a question I get from a lot of people. And I would say, I hope so, but the evidence is very limited. So in our original study, or in the second one, I can't remember offhand, in the second paper, I think we have a graph where we show the response to growth hormone and whether the patients have been treated with either a GnRH agonist or an aromatase inhibitor. And there were, to my knowledge, I think 2 patients who were treated with an aromatase inhibitor, which seemed to delay their bone age advancement. However, both of those patients, they were siblings, they were not particularly compliant and were lost to follow-up. So we don't have any final outcome data on them. I know of a number of patients now around the world who are being treated with aromatase inhibitors, but to my knowledge, there's no kind of control data or, you know, even published longitudinal data of a patient with a clearly pathogenic Aggrecan mutation being treated with an aromatase inhibitor. I personally haven't encountered yet a child who's entered puberty who needed that under my own care, but that is definitely something I would consider for those patients. Then I would monitor the bone age to see whether it seems to be having an effect on slowing the bone age advancement.
All right, thank you. A question from Ada Sola, Ada Koya. For the children delivered SGA, at what chronological age was the bone age done?
Oh, in that particular paper, so that was a paper out of the Netherlands, and I do not remember offhand when. I think that what happened was, I think that was a retrospective look where they looked at kind of all of they have a large cohort of patients with who were born SGA, and I think they just looked retrospectively at anybody who had had an advanced bone age at any point in time, and then took those patients and sequenced the Aggrecan gene to look at the yield. So it wasn't a standardized assessment, but what the point that you're bringing up, which is a very good one, is that the bone age advancement may be more subtle in the younger children, although I've seen some young children with quite advanced bone ages, and it might progressively worsen over time. So it may not be apparent, you know, in the first few years of life.
Alexander Augusto de Jorge, he says, congratulations, Andrew, for the comprehensive and wonderful lecture. Thank you. He does have a question. He says, is the analysis of epistasis between variants common, rare common variants, the next step to better understand the genetic architecture of height variability?
So it's a great question and a tough one from Alex. So I should say, you know, to give credit where credit is due, Alex is one of also the leaders in this field and has found the paper that I alluded to about Indian hedgehog, and some of the NPR2 papers are from his group. So great work there. So Alex is talking about the interaction between common genetic variants and rare variants. And I would say that, yes, that's definitely an area that we have to go into more. And there are, in other disorders, there have been, like for example, in hypogonadotropic hypogonadism, there have been studies looking at pathways of genes to see whether if you have multiple hits in a pathway, whether that increases your risk for a certain disorder. And I think what we're going to find is something similar in short stature, where you might have multiple common variants or a common and a rare variant working together. The problem is statistically to prove that for any individual variants is exceedingly difficult. I think that, experimental models will be needed for that. So I think that's one definite area for future research. I also think that epigenetics has a lot of potential area for research as well in growth.
Wonderful. Gada Anwar wants to know, a height below 1.6 SDS is considered short stature and requires investigations?
No, not necessarily. Definitely not. There's always going to be a normal range for individuals, and that's determined statistically, right? So 2 standard deviations represents 95% of the population. So I definitely wouldn't raise that, but I would say what you need to consider is each individual patient and their growth pattern and where they are with regards to their family. So it's possible that somebody might be minus 1.6 standard deviations, but everyone else in their family is plus 2 standard deviations. Well, in that case, something's probably going wrong, and that patient might require further investigation. But even I wouldn't say that children below minus 2 necessarily need significant evaluation. It really depends on the family context and the growth pattern, and whether the growth chart is appropriate for that individual patient's ethnic background as well. That's a problem we often have in the United States, where we apply a single growth chart to people who come from all over the world, where that growth chart might not be representative of their own backgrounds.
Thank you. Susan Shred, she wants to know, how can Vosoritide overcome the dominant receptor mutation?
Good question. So I think you're talking about Vosoritide and the heterozygous NPR2 mutation. So the idea there is the dominant mutation that is actually, you may be alluding to, it has what's called a dominant negative effect, where you need 2 copies of the NPR2 receptor and they interact with each other. But what's going to happen is sometimes those 2 copies will be healthy copies. Sometimes those 2 copies will be 1 mutant and 1 healthy, and sometimes it'll be 2 mutant. And when it's 1 mutant, 1 healthy, it impairs it, but it doesn't totally knock out the function. And the idea is that if these patients have 50% of the normal function, or even less than that, 30% of the normal function, if you can increase the signaling coming in and raise that to 75 or 100% even, that could restore the normal amount of CNP signaling to the chondrocyte. And there is some data, some in vitro data to suggest that that is true.
Ola Nielsen wants to let you know, congratulations to a wonderful lecture. She also asks, can you elaborate on the genetic explanation to the relatively large variation in adult height within families plus minus 1.5 SDS?
Oh, so that's a good question. So again, giving credit, Ola led a lot of the Aggrecan work, so he's been a real leader in this field as well. Ola, I don't know that I have a great answer for that. I've always assumed that, it is just the aggregate difference in the polygenic risk that occurs, within an individual family, and that while we think about just the common variants, that there are a lot of these moderate effect variants as well, and that if you just get a few of the moderate effect variants, that could, lead to a wider range of heights within an individual family. But I don't really have any other specific thoughts or answers to that question.
Thank you. Leanne Ward has a question. Is there any experience with patients who have ACAN mutations experiencing a worsening of their osteochondritis dissecans? And forgive me for mispronouncing. I have a patient who seemed to deteriorate WRT to OCD following GH therapy in this setting.
Yeah, that's also a great question. So not that I know of, Leanne. What we don't know is that an effect of the growth hormone on the osteochondritis dissecans, right? Or is, or is that just the natural history of that child's underlying osteochondritis dissecans, which in patients with Aggrecan can be quite aggressive. In our study thus far, we have not seen a worsening of the joint disease in those individual patients, but it's a very small number of patients. And of course, there's no control group, so we really don't have a way to answer that question. I'll tell you what we're counseling them. We're having them see a sports medicine physician and orthopedic doctors, and we're counseling them to avoid higher impact activities, which could potentially worsen the arthritis. But we do not know the answer to that question yet.
Thank you. Guillermo Alonso has a question. Is puberty advanced in patients with ACAN mutations? Is it according to the bone age or chronological age?
No, so puberty timing seems to be normal and just dependent on the typical factors that affect, pubertal timing in any individual family. And it does not correlate with the bone age in Aggrecan mutations, which is one of the key features of it.
Thank you. Luiska PC has a question. First of all, she says, thank you for a very nice review. Can you comment on the social implications for patients with achondroplasia who think that Vosoritide therapy will make their children feel inappropriate, as commented on the New York Times article from this weekend?
Yeah, I read that article. I think that's a very interesting article. So again, just to be very clear, I had zero involvement in that study. And personally do not treat any patients with achondroplasia. So I don't know that I'm the best person to comment on that. I think that's a really important issue. I think that, it's an important issue not just for patients with achondroplasia. I think it's an important issue that we need to think about whenever we're treating any individual with a medication for their height. And personally, in talking to families, I think that families come with very different views on this issue. Some families really think that height treatment can have a significant effect on children's quality of life and attitudes and, ability to succeed and thrive. And other families feel exactly the opposite. So, as a practitioner, I just try and be sensitive to those issues and kind of present it with an open mind. I think for achondroplasia specifically, the hope, and this hasn't been shown yet, but the hope is that if started at an early enough age, Vosoritide and other CMP medications may also be able to alter some of the other significant medical comorbidities that occur, not just have an effect on height, but have an effect on other medical complications that occur. Now, we'll have to see if that turns out to be true over time, but I think that's an important issue to consider as well.
Wonderful. Thank you. So we have time for just one more question. That question comes from Samay Tafik. He wants to know, thank you, first of all, thank you very much for the excellent presentation. When should we request FISH in Turner syndrome?
Great question. So, there are guidelines that came out in general with regard to Turner syndrome, suggesting that any girl who has unexplained short stature should have a karyotype or, have karyotype done to look for Turner syndrome mosaicism. I would say what's been shown to be one of the best clinical predictors, though, is the distance in the child's height from the midparental target height. So we did a study looking at patients whose height was below -2 standard deviations, and they had a height of 1 standard deviation or more from their midparental target height, and found that that was a high risk group for having, undiagnosed Turner syndrome or Turner syndrome mosaicism. And other groups have also done with various cutoffs of the distance from the midparental target height with or without short stature. But I would say, you could always draw a line somewhere, but the clinical take home there is if that child's height is different than if a girl's height is different than the midparental target height, I would have a very, very low threshold for ordering karyotype for testing them for Turner syndrome, since there are such important other health implications and fertility implications for that child, that I would err on the side of over ordering that test.
Thank you so much, Dr. Dauber.